With the advancement of medical technology, new treatment methods have emerged for cancer. In this episode of Innovative Breakthroughs, we've invited international experts to unveil them for you. This episode is sponsored by BeiGene. 欢迎收听这一集的创新突围，我是主持人 Ethan 刘杰忠。今天很特别因为是我们第一次采用英文特辑的方式来制作，而且是跟医学相关的内容。一方面，后疫情时代大家对健康议题特别注重，再来是癌症已经是仅次于心血管疾病全球第2大的死亡原因，所以今天跟大家分享一个令人振奋的治疗方式，叫做 cell.therapy 细胞治疗，可以说是病患还有家属的新希望。今天特别邀请到2位国际的专家，他们一起聊一聊细胞治疗的现况、趋势以及展望。Hello and welcome to Innovative Breakthroughs. My name's Ethan. Thank you for joining me on today's podcast series with 2 international experts as we explore the topic of cell therapy as a breakthrough in cancer treatment. So let's welcome our special guests. First of all, we have Professor Stephen Opat from Australia.
Thanks, Ethan. Hi, my name is Stephen Opat. I'm a haematologist, a blood cancer specialist. I work at Monash Medical Centre, which is a large university teaching hospital in Melbourne in Australia. I have a particular interest in the search for new therapies for patients with blood cancer and have been running clinical trials for over 20 years. So my hospital currently has over 60 trials open just in blood cancer. And I'm over here in Taipei visiting the BeiGene cell therapy facility. So it's very exciting to be in Taipei and look at the next round of therapies for patients with blood cancers.
Also here with us today is Alex Huang, his vice president at BeiGene, which is a biotechnology company focused on the development of innovative and affordable medicines for cancer treatment. Welcome.
Hi, Ethan. So this is Alex. I'm the head of cell therapy at BeiGene. Before BeiGene, I was with Novartis, Sanofi, Genentech, BMS, and AbbVie, leading teams and advancing programs from target ID validation to preclinical studies, IND, phase 1, phase 2, phase 3, and lifecycle management. It's great to be here.
All right. So first thing first, let's talk about cell therapy. For some people, like me, myself, before I met you guys, it sounds like a marketing gimmick, a really big fancy term printed on the bottle of a skincare product. But no, we're all sitting here. It's the real deal, right? So tell us, how does it work and how is it different from existing cancer treatment options?
So previously, when we're treating cancer, we either had radiation therapy or we had drugs or chemicals which target different processes within the cells and kill the cells. So that's sort of traditional chemotherapy. Then there came immune therapy with antibodies. So antibodies are part of the immune system that bind to the tumour and direct the patient's immune cells to find the cancer cells and kill it. And the next innovation was the use of cell therapies. Now, cell therapies is not that new. The first bone marrow transplant was done in 1956. And originally, the idea of bone marrow transplantation was that you could give a big dose of chemotherapy or radiation that would destroy the patient's bone marrow where the blood cells are made, and you would rescue them by infusing bone marrow from another person. But it was later realized that the other thing you're doing, you're infusing the patient with immune cells. This is getting around to this topic of immune cells, which then can find the cancer and kill the cancer cells. And that's called graft versus tumour effect, where the donor immune cells, mainly lymphocytes, kill the tumour. After the realization that you can use cells to kill cancer, the scientists, back in 1993, the first one, they said, well, maybe we can re-engineer these immune cells to fight the cancer better. And that's what we're here to talk about today. Initially, there are some already approved products called CAR T cells, where the patient's immune cells taken out of the body, they're manipulated or re-engineered to fight the cancer, and then they're reinfused back into the patient. So while the first CAR T cells were made in 1993, the first successful treatment in 2019 in blood cancers in lymphoma and leukemia.
So it's like you take out those cells and send them to boot camp and train them and put them back in the patient's body to attack cancer cells. Can you say that?
Pretty good, Ethan. That's pretty good.
You've got it. Do you want a job? Yeah, you can do it. So you can run the boot camp. So what you do is you reprogram, you change the genes, the DNA, the genetic information, and you say, you're not an immune cell to fight the common cold. We're going to retrain you to fight lymphoma cancer. So you put in a gene that makes that immune cell, which could have been responsible for fighting something else, but you're saying, now you're not a common cold immune cell, you're a lymphoma killer immune cell, and you put in a receptor for something that's expressed on a, say, lymphoma cell, and then that immune cell, when it's reinfused, will circulate in the body, will find the cancer and it will kill it.
Okay, so you mean the receptor is like a key, right? Because any one key can only solve one lock, right? So you inject one specific receptor on the cell to attack a certain type of cancer.
Yep. And the key, so the commercial CAR Ts, the first target was a thing called CD19. CD19 is a protein expressed on lymphoma cells, mainly. You want a key that's specific. And so they get these cells and they put in a receptor with signaling for CD19. And then when you put the cells in, they will find the tumor expressing the CD19 protein and kill it. So that's sort of where conventional CAR T cells, how they work.
Yeah, but I think no discussion about cell therapy would be complete without the story of Emily Whitehead, right? The case you have just mentioned. I think she's the first pediatric patient to ever receive this CAR T cell therapy.
Yeah, I mean, and I think that, she's definitely one of the very good examples, successful examples. But I do believe that Stephen actually has also his personal experience as well. And I thought those are really nice examples as well. Yeah. Stephen?
I have a patient I looked after, a 37-year-old man with 3 children. And he had lymphoma, diffuse large B-cell lymphoma, which is one of the commonest forms of lymphoma. And he had 7 different therapies, including a stem cell transplant, and failed all of them. And then he went on 2 trials with some experimental drugs and progressed. And then he got these CD19 CAR T cells in October 21. And as of today, nearly 2 years, he's still in remission. So he'd failed 7 or 8 lines of therapy. And usually in aggressive lymphoma, if you're in remission after 2 years, it's unlikely to come back. So we never use the cured word, but we're very optimistic.
Yeah, I mean, it's actually, hearing this kind of stories every time, this is what, motivates us. I mean, Stephen, I'm pretty sure, in your hands, you have saved a lot more patients. But to hear this kind of story, this actually makes our day in terms of in the drug discovery and development, because this is what we work for. This is what we work towards.
Yeah, I think Professor still has the smile on his face while he was talking about the story that happened 2 years ago.
Yeah, we're very happy for the patient, but you've got to be careful as a doctor. You don't feel like you're a Superman, like a superpower.
Okay, but from stories from Professor's case, are you saying that the cell therapy is usually the last resort in terms of cancer treatment?
So any new or experimental therapy, you leave it until you've used all the conventional therapies. So it used to be if you fail chemotherapy, then a stem cell transplant was the next best step. And as therapies get more established, they move earlier in the course of treatment. So we know about cell therapies that if you give them earlier, they're more effective, because after you've given the patient a lot of treatment, their cells can be quite sick, which this will be an important, I know Alex is going to talk about allogeneic CAR therapy. And one of the issues why we need allogeneic CAR therapy is because if you've given a patient 6 or 7 different lots of chemotherapy, their immune cells they're struggling. And so to get them to kill a cancer is harder because they've been damaged by all the previous therapy.
Okay. So you were talking about this patient who has lymphoma. So I think our audience must be curious to know what types of cancer can be treated by this CAR T cell therapy, lymphoma or other types?
So there's established treatments, and then there's our what we only can imagine. So the established treatments at this stage are lymphoma and lymphoblastic leukemia. So in a lot of countries, the CAR T cells are reimbursed for these conditions and they become standard of care. But, there are many other diseases. Really what would be great is if we had treatments for like pancreatic cancer or brain cancer. And I think the blood cancer story is just showing the proof of concept that this can work. Now we need to work out how to adapt it for other forms of cancer. And that's a lot of work, but will be life-changing for many people if we can. Blood cancers are only 12% of all cancers. So 88% of cancers are not blood cancer. So a lot of work to do.
And, maybe I can chime in a little bit here. I think that what Stephen mentioned earlier, in the cell therapy, we can make meaningful genetic modifications to strengthen the cells. It's like what you said, right? You take the cells, you go to boot camp, make them stronger. Exactly that. Yeah. So we know that solid tumor, the environment around solid tumor is a little bit different from environment in blood cancer. And we also know that in order to overcome those environmental challenges in solid tumor, we do have to make modifications to these cells that we want to make them stronger, make them live longer, just so that they can actually tackle or target the solid tumor as efficiently as they are in blood cancer. And so this is definitely something that we're striving for still.
So unlimited possibilities, right? We can all look forward to. But Professor just mentioned another concept I'll try my best. Allogeneic. How do I sound?
Perfect.
That's perfect. Yeah.
Allogeneic sounds that the cells are taken from the patient's own body, or no?
So allogeneic, so autologous is from the self, from the patient, and allogeneic is from a donor that may be related or unrelated to the recipient.
So most CAR T cell therapies to date are generated using autologous
Perfect,
cells, right?
perfect. You
got
it right.
Yeah, you got it.
Yeah. But I know BeiGene has been working on another approach, right? Since not very long ago, 2021.
Yes,
right?
yes.
Why? Tell us more about this.
Well, so I think that for the autologous CAR T therapy, I just want to make this straight. I just want to make this point up front, right? It really brings tremendous value to patients. That's undeniable. Some of patients are even considered cured. The problem is that, we kind of touch upon this a little bit, is that because the starting material is taken, the cells are taken from patients, and already these patients might have gone through several rounds of different treatment, their immune system might be compromised a little bit, and therefore your starting material might not be as good. And therefore, you could imagine some patients might have a better immune system, but not the others. So during the manufacturing process, right, the final product you get really depending on the quality of the starting material. And then there's a variability in terms of the health of the immune system from different patients. So you could imagine how that could actually throw a lot of hurdles to the manufacturing process. And inevitably, that would make it expensive.
How expensive?
So in US, it's close to $500,000, $500,000.
Wow.
And that's a lot. That's a lot of money. And therefore, not everybody can afford it. And that's why people start to think about allogeneic, meaning that I can go to healthy donor as a starting material. And if I make some changes to these material starting cells from healthy donors, I can probably still put it into patients. Even they're coming from different bodies, if you will, and that would still be effective. And also at the same time, by definition, I'll be able to mass produce it and essentially becomes out of the shelf, right? So meaning that you can just take it from the shelf and then administer it. So this will be a lot easier and will actually reduce a lot of costs. This is what we are working on, basically.
The other point I wanted to add is off the shelf, it's fast. If you've got allogeneic product on the shelf, you can give it immediately. So the problem with autologous CAR Ts, you have to ship the cells off, often to the US or to sometimes Singapore, and then they have to be manufactured, they have to be manipulated, and that can take a few weeks. And in patients with bad cancer, they might not have time to wait for the manufacturer. So we're hoping that allogeneic CAR cells will enable a faster delivery, less waiting time.
It's like you're talking about something that's tailor-made, right? If something that's tailor-made, it takes time and money.
Exactly. And also at the same time, I think there's also one more thing I just want to kind of mention. So the allogeneic, and we talked about the starting material from donor, for example, right? So that's one approach. Another approach is also from donor, but now what I do is I take the donor's cells and I make it into an induced pluripotent stem cell state, meaning that, with this kind of approach, now you have a chance to have the starting material all coming from the same cell clone. And that could even dramatically reduce the cost of goods even further and reduce the heterogeneity. And essentially, it would ensure the quality and the homogeneity of the drug, as well.
Okay, so basically our experts are saying that allogeneic cell therapies produced in large batches have the potential to address the problem, like logistics problem and the manufacturing costs, right? But it's not without its challenges, right?
You totally get it. I think you should change your profession.
I got a job opportunity here during the interview. Yeah. Talk about some challenges or hurdles that we face.
So the first is the cost. And Alex mentioned the cost of the drug, but there's also the hospital cost, the cost of supporting the patients, which can be hundreds of thousands of dollars. Then there are two complications of CAR T cells, one called cytokine release syndrome, where you have an inflammatory immune response and the patients get high fevers and they can get very sick. They can even need to go in intensive care. And then there's another syndrome called ICANS, which is this neurological syndrome. So, when people get fevers, they can go off neurologically. And so the patients can get drowsy, they can get comatose, they can have seizures. All these things are very manageable, but it means that you need to have centres that are experienced in delivering CAR T cells, know when to give the antidote to prevent these severe reactions. And we're hoping that some of these allogeneic CAR Ts may not have these problems which the autologous ones have.
Mm-hmm. So Alex, did you observe any hurdles or challenges in addition to what Professor has just contributed?
Certainly. And as I mentioned earlier, the autologous CAR T, this is like the first generation. It already does a lot of good for patients. Our next challenge would be that how can we make allogeneic CAR T therapy even more potent, even more efficacious, and even with more persistence, right? In order to essentially capture all the advantages of the autologous CAR T and then some, basically. And I think that actually requires a lot of understanding about our immune cell types, about our immune system, how to modify them, for example. Are there any components we can add to these cells to make it stronger, to make it live longer, right? It could be through genetic knockout, meaning that you can modify genetic content, or you can add on things, basically. And so these are all what we call the cell therapy engineering, right? So you're really engineering, essentially, a certain cell type of interest into a very robust therapeutics. And these actually would require years of work, right? First of all, you have to have a very good, I would say, fundamental understanding about the basic biology, but also at the same time, you got to develop the tools that can do the gene editing efficiently. And these would all contribute to the final product of the allogeneic cell therapy. And so, these are a lot of these challenges and hurdles that I just mentioned. There are so many people working on it at the same time. It's a big scientific community. And I do believe in the near future, there will be breakthrough. And actually, there are breakthroughs already that we will be able to overcome these challenges and really create a product that would actually be life-saving.
Okay. So let's talk about the progress of autologous T-cell therapy. Is it getting more and more common, or it's still in its early phase?
It is more common. So at the moment, I'll talk about the situation in Australia. There is approved products for treatment of lymphoma in second relapse, so as the third treatment, and also in younger adults with acute lymphoblastic leukemia. So if you have one of those diseases, then there's a committee that's been set up by one of the government agencies, the Medical Specialists Committee, that reviews all cases and they say, yes, this patient is suitable for CAR T. They've got to be, have sufficient organ function that they can walk around. They can't have an overwhelming infection. So they meet a number of criteria. Their kidneys and liver function have, if they meet the criteria, then they're individually accepted on the program for funding. And then there's a number of authorised CAR T delivery centres, and so they will be slotted in. But there's a lot of diseases where CAR T's therapy has been shown to be of benefit, such as mantle cell lymphoma and follicular lymphoma, but these are not yet funded. And I think the major problem is really the cost is enormous. I know it's a small population, but I think this is why we're hoping that allogeneic CAR T, it will be more of a reality. And, Australia, I don't know, is a fairly wealthy country and can afford this, but the main issue is that most of the world won't be able to afford, the standard autologous CAR T. They're too expensive.
So far, allogeneic CAR T cells are still beyond ordinary people's reach, right?
Well, I mean, right now, I think that there are several, I would say, clinical products that's in experimental stage, meaning that they are in clinical trials. Certainly, I think that, there might still require some time to bring it to commercialization. But this is what that we all been working towards, is to bring the first allogeneic product, a cell therapy product, into commercialization.
Yeah. What's your expectation?
I expect a lot.
But what's the time frame we're talking about here?
I would say I would not be surprised if in the next 5 years there's actually a major breakthrough. Usually, if there is a major breakthrough in a clinical trial setting, it would accelerate. Because a lot of regulatory bodies, they see that the data are so dramatic, good, they would accelerate it, meaning that they will have an accelerated approval, and that means that this drug would get a chance to be to have access, to let patients to have more access from the commercialization perspective. And I would not be surprised if in the next 5 years, allogeneic cell therapy would achieve that status. But, Stephen, please feel free to chime in.
So if I look at what's happening, there's now trials that show autologous cells are good second line. So I think the regulators will take a while to catch up. We're going to see autologous cells used in patients who fail the initial therapy. So I think that will be a game changer. The other thing, there's even trials going on looking at these cells as the initial therapy in patients with bad risk disease. So I think the majority of patients do very well with chemotherapy, so it's going to be hard to make a cost-effective argument with an expensive treatment, but we'll see what those trials show.
All right. So for the final part of today's program, I want to talk about some legal aspect, because a cell therapy, where we talk about gene modification or re-engineering, that sounds really exciting, but we should remember that innovation and regulation are two sides of a coin. So Professor Opat, you're from Australia. Can you tell us more about this regulatory environment for cell therapies or any new medical innovation in your country?
It's a complicated question because there's two parts. There's, first of all, the regulation regarding a genetically modified organism. And so whether it's a cell or a bacteria or a vegetable, there is a statutory government body where you have to get a license for any genetically modified material. The main thing they want to check the biological safety, that it's not going to escape and cause some problems. So the process is straightforward. You just apply for a license and then you get a license. So once you have a licensed product, then you work with the TGA, or the Therapeutic Goods Administration, to run a trial. Once you've got a license, it's fairly straightforward. You have a trial protocol and they will review it, and then they will give you a permission to go ahead, and then you conduct a trial. Once you've the successful trial, then there's a whole other complicated process about applying for registration of your product. And then after that, then there comes the reimbursement, which is, the very last hurdle. So for instance, there will be autologous CARs. I can tell you now, there's an autologous CAR approved for mantle cell lymphoma in Australia, but it's not reimbursed. So if you can afford $500,000 US, you can get access to the car. So I think the government will eventually come to fund it, but they haven't made that decision yet.
But it has to go through like more steps than regular therapeutic goods, right? But I think Australia is already known for its abbreviated regulatory pathway, right? But do you have that optimism in other parts of the world, Alex?
Yeah, I do. Even though we got to recognize that, with any of the new therapeutics, like cell therapy, it's going to take a while for the pharmaceutical industry and the regulatory bodies to interact. It's almost like a mutual learning, right? So for the regulatory bodies, the cell therapy, especially the allogeneic with different sources, these are still quite new in a way for the regulatory bodies. So in a way, I think that we have to learn from the regulatory bodies in terms of the safety concerns, in terms of what they look at. And of course, we want our medicine to be as safe as possible. And all the same time, I think that the regulatory bodies are, they're also learning from the pharmaceutical industry in terms of what are the essence of this new therapeutic modalities. So I think it's an interaction. And usually by these interactions, then more and more we come to a very, I would say, rational assessment of every new cell therapy programs that come into the clinical development, right? And I think that this is no different from I would say years ago for the small molecule clinical development or for the first biologics clinical development. There's no difference. Essentially, we all have this kind of a very interactive conversations through the regulatory discussions, and then it is through that and with time that we'll gradually build up a very efficient process for their approval.
Yeah. And speaking of interaction, I know I was told that BeiGene just set up a research facility in Taipei where Professor Opat visited for the past 2 days, right? How was the trip?
Oh, incredible. I mean, what was really impressive was this research laboratory is only 18 months old. It looks like it's been there for years and years. It's full of scientists, full of equipment, and they're producing candidate cells already. I don't know how they've managed to do things so quickly. So, well done to BeiGene for getting things, and maybe it shows how important it is to them, how much investment they put behind it, so that they're really trying to get things to market quickly.
Yeah, thank you, Stephen. I think there's a saying in BeiGene. It's called BeiGene speed, right? It's
faster
quicker
than light speed.
It's quicker than any of the fast you can imagine. Yeah. But I have to say, I mean, the motivation behind that is really that we want to, if we believe in our medicine, we want to try as hard as we can to get that medicine, experimental medicine, to patients. Right?
They're also running with time, right?
Exactly. Because what we believe is that every little effort that we put in could save potentially a lot of lives, right? And so in that consideration, we believe we should throw in everything we got and move as quickly as possible. And I think that's the motivation behind the so-called BeiGene speed.
And it's not just a cell. We've dealt with other BeiGene development, and every phone call, can you put more patients on the study? Hurry up, and always wanting all the stuff done yesterday. So the culture of the company, I think, is really applying a lot of pressure for people to respond quickly. So I think it's paid off.
But it was just set up 18 months ago?
Yeah.
During the pandemic?
Yeah, and so I mean, what's more amazing is that because we have to build everything from scratch. There was no lab, there was no office space, so we actually did all that did all the constructions, and also building up the team, meaning recruiting talents and starting doing experiments, all done at the same time in parallel. I'm really grateful for the team's relentless planning. There's a lot of hard work going into it. There's a lot of well-designed, organized scientific exploration, and precise execution as well. And that's how we try to, I would say, live up to the BeiGene speed.
But why Taipei in the first place? Have you thought about the options?
I think that, a lot of people ask me that question, especially when I first, came to Taipei and established this cell therapy research center. And my answer is always, why not? Taiwan has such a great research talent pool. There's Academia Sinica. There are several, numerous first-line universities. There's a great medical research. It's been a pretty major APAC clinical trial site as well. So essentially, Taiwan has all the things that could make the drug R&D successful. So my answer to that question is always, why not? Why not? And to be honest with you, regardless of my answer, what we have achieved so far is a testament to why not. And I'm hoping that starting from us, no one's going to ask me anymore, why Taiwan? Okay?
All right, this deserves a great round of applause.
And then everybody would just say, oh, yeah, that really makes sense.
Yeah. All right, so Professor Opat, that brings us to the last question. What's your vision, or how do you foresee this development of cell therapy, or in particular, this from the donor's product? How do you see this trend?
I think we're at the start of the journey. We've really, the early products, which have been around since 2009, they're really just the start of the journey. And I think I'm hoping that, one, that they will be available everywhere, not just at a few centres globally, and two, that they will come down in price. And I see them moving earlier. Who knows? There's a lot of diseases where chemotherapy is no longer given. So the disease, I treat a lot of chronic lymphocytic leukemia. We don't give chemotherapy anymore. So I'm hoping that we can treat cancer being smarter, giving these immune cell therapies. We might, if they were cheap enough and the trials are positive, we might even be able to give them in the front line. And so when people have these devastating cancer diagnosis, then they would just, they have to take the day off work, get the cell therapies, and they go back to work on Monday, all better. That would be ideal.
That would be wonderful. That would be wonderful.
But before that happens, do you have any advice for patients or their families?
So my main advice is, I think if you have the ability to get involved in any clinical trials, I would do it. That often people are a bit apprehensive of new therapies, but in the main, patients who go on trials have better outcomes than patients that don't go on clinical studies. And the only way we're going to be able to get these new drugs if they can be tested on the patient. So we ask the patients to trust us, and that often there's a lot of thought, a lot of smart people developing these compounds, and there's a lot of scrutiny and regulation. So in general, you won't be able to give a therapy that's particularly toxic. An ethics committee would have stopped it happening by then. So I think the one bit of advice to our patients is things look really good, and definitely if there's a clinical trial for your condition, no matter what it is, I would definitely encourage participation.
So Alex, what about you? What's your maybe goals in the near future, maybe for your company or for the industry as a whole?
Well, I think that, as I mentioned, on the cell therapy front, it's a big scientific community. We're very happy that we are contributing to the scientific community. And I think that in the near future, we're hoping that we'll contribute to the medical community as well and bring hope to patients. And we are working towards that as quickly as we can with BeiGene speed.
All right, so we look forward to more good news. From today's discussion, cell therapy offers a new approach to fighting cancer. It holds great promises, yet it's not without challenges. Meanwhile, it requires a strong, innovative power and collaborative mindset of the government, pharmaceuticals, and medical industry as a whole. We look forward to its future directions and developments. It's a quite honor today to have Professor Stephen Opat and Alex once again. Thank you both for joining us.
Thank you, Ethan. Great to be here.
Thank you, Ethan.
感谢您收听创新突围的英文特辑，我们下次见，拜拜。